And I'm sure that you are very familiar with the terms I've just mentioned. Uh, TO, Stoxo, R is rubella, German measles, right? Cytomegalo, not a very nice thing to have.
Yeah.
And we look after, especially, we're very careful about blood transfusions. And H is herpes. We've got HSV 1 and 2. And HSV is not a very nice thing to have, herpes. You can have genital herpes, you can have herpes on the skin, and it's not a very, very nice thing to have. Hi, Nazneen, you made it. Nazneen, unmute. Chal se chalo.
Hello, good afternoon,
Hi,
ma'am.
good afternoon.
Hello, ma'am. Good afternoon. How are you?
We are all okay. We're just
How is
talking.
everybody?
I'm just asking these gals if they know about the torch test, and they said they do.
Ma'am,
Huh?
what is this MU capture you have mentioned?
Huh?
MU capture.
New capture for recording to what?
Regarding this toxo IgG and IgM uses MU capture. Can be done using ELISA, HLA.
Yes. That is the methodology. Let me just go through it. The different types of methodology. We can do it with ELISA. We can do it with uh, other machines are there. You know. So using the other principles, and they are more specific. So shall we start? Right? So get the first one. You all can unmute. Okay, so the title is Significance of Torch Tests. Alright, let's go to the first slide. Alright, so we've got this little torch. On one side we've got viruses, because the others are viruses. The cat parasite is there, and we're going to talk about a mixture of all this. So what is the torch panel? So it stands for toxoplasmosis, rubella, cytomegalo, and herpes 1 and 2. The combination may be IgG, past infection, or IgM, which is the present infection. Hmm? So you can we call it torch 4 and torch 8. So if it's torch 8 means both IgG, IgM is asked for. When they say torch 4, then we ask what do they want? What do they want, IgG or IgG? They want IgM. So the question is, what is toxoplasm? Toxoplasmosis is an infection that can threaten the health of an unborn child. It is caused by the parasite called Toxoplasmosis gondii. The parasite multiplies in the intestine of cats and is shed in cat feces. One can also get the parasite from eating undercooked meat, such as rare beef, from animals infected with the parasite. If one gets toxoplasmosis for the first time, when one's pregnant, or up to 3 weeks before one conceives, there's a risk that the infection may cause an abortion and can also affect the health of the baby if the baby is born. So not a very nice infection to have, isn't it? All right. So signs, symptoms, and prognosis. During the first few weeks of post-exposure, the infection typically causes a mild flu-like symptom, no illness. Thereafter, the parasite rarely causes any symptoms in the otherwise healthy adults. However, with those with a weakened immune system, such as AIDS patients or pregnant women, may become seriously ill, and it can occasionally be fatal. The parasite can cause encephalitis, inflammation of the brain, and neurological diseases, and can affect the heart, liver, inner ears, and eyes. Recent research has also linked toxoplasmosis with brain cancer. My God, look, it spreads all over. Okay. So why and when is it important in pregnancy? That's the question we ask. If a woman presents a history of abortions or repeated abortions, this test becomes important. When can the test be done? Toxo IgG and FIGM uses mu capture, can be done using principles of ELISA, ECLIA, Roche E411, Alexis using ECLIA technology, latex is also used. I think mu is something to do with latex. I'm not very sure. I'm not very sure of that. But these are different methodologies, and so you need machines for it. You can ELISA machine is quite okay. Your ECLIA machines are, they are quite fancy. Can it be treated with drugs? Of course it can. Like azithromycin dose pack. The generic name is azithromycin class, macrolides. Bactrim, generic name sulfomethazone. Then you've got trimethoprim, miscellaneous. And you've got Biaxin XL, generic name, clarithromycin class, macrolides. So there are drugs that can be that is given, and of course it also depends on your doctor how much is given and when. Okay. Advantage Roche toxo assay using Vicobas E411. The kit does not pick up or detect persistent IgM. Therefore, less or no cross reactivity or interference in assays like others, which then requires retests. It's a mu capture assay for toxo, like cytomegalo and herpes. I think mu is it is something that is present on the parasite itself or on the virus itself. It's a double antigen sandwich assay for rubella. Assay time 18 minutes per test. Most of these Roche assays are 18 minutes and quite accurate. Roche ECLIA assay for the detection of toxo IgG and M using E411 and Alexis platform. It's an accurate, sensitive and specific assay using a 2-point calibration method to calibrate the test. So now we are asking a question, what is a virus? Because the cytomegalo and all are viruses, as are rubella. So virus particles known as virions consist of 2 or 3 parts. The genetic material made from either DNA or RNA, long molecules that carries genetic information, a protein coat that detects these genes, and in some cases an envelope of lipids that surrounds the protein coat when they are outside the cell. Just point it out. Can you point it up there? इसके ऊपर. Take a look. The shapes of viruses ranges from a simple helical or isohedral forms to more complex structures. The average virus is about 1/100th the size of the average bacterium. Can you imagine how tiny it is? Prions are infective protein molecules that do not contain DNA or RNA. So take a look at the structure. You've got the things in the middle and you've got the coat. No, don't take it off. Can you see this? All right, let's go to the next one. All right, now the complete virus particle, known as the virion, consists of nucleic acid surrounded by a protective coat of protein called the capsid. These are formed from identical protein subunits called capsomeres. Viruses can be a lipid envelope derived from the host cell membrane. The capsid is made up of proteins encoded by the viral genome, and a shape serves as a basis for morphological distinction virally coded proteins. Above picture is an influenza virus. Take a look. I'll hold for some time. So you've got the protective coat called the capsid. Then you've got an identical protein subunit called capsomeres. You have viruses can have a lipid envelope, and so on. All right. Oh, that's done. All right. So we are asking a question about the genome, and I think you've heard of it for many years now. The whole study has been done. The cartoon says God, the human genome codes been unraveled, and you said God saying, "Damn hackers! Now I have to change the password." So people have gotten and found out what the genome is all about. Genome is the entirety of an organism's hereditary information. It is encoded either in DNA or, for many types of viruses, in RNA. You know that there are RNA and DNA viruses. The genome includes both the genes and the non-coding sequences of the DNA RNA mixtures. All right. So that since we spoke a little bit on the virus, we take the first one, TOR, T-O-R, rubella. The disease is caused by rubella virus, a tagovirus that is enveloped and has a single strand RNA genome. So it's an RNA virus. The virus is transmitted by the respiratory route and replicates in the nasopharynx and lymph nodes. The virus is found in the blood 5 to 7 days after infection and spreads throughout the body. The virus has teratogenic properties and is capable of crossing the placenta, infecting the fetus, where it stops cells from developing and destroys them. So the baby is involved. Increased susceptibility to infection might be inherited, as there is some indication that the HLA-A1 or factors surrounding A1 on extended haplotypes for involvement in virus infection or non-resolution of the disease. So you've got a picture of the rubella below. I don't know whether you all have had the rubella infection. I've had it very late in life, and I was surprised when it came on. Lasted a few days, redness, a little bit of bone pain and stuff like that, and a little bit of these little red spots all over my face and hands. Okay, next one. Why is it considered dangerous in pregnancy? If rubella is caught within the first 3 months of pregnancy, it can damage 90% of unborn bodies, including eye problems such as cataracts, cloudy patches of lenses of the eye, deafness, heart abnormalities, and brain damage. So it's quite potent. Why and when should the test be done? When a pregnant woman is suspected of having German measles, this test must be done immediately, as it may affect the child badly. What are signs and symptoms? The rash or rubella on skin of child's back. Distribution is similar to that of measles, but the lesions are less intensely red. Take a look at that picture. Okay? What tests are to be done on such a patient, especially a pregnant woman? Yeah, of course, it's asked for rubella, IgG, IgM. Methods: ELISA, ECLIA, and on the principle of ECLIA. Okay, they're using different machines. What is cytomegalovirus all about? Cytomegalovirus is a double-stranded DNA virus and is a member of the herpes viridae family. Other family members include herpes simplex virus, HSV1, and herpes simplex virus HSV2. So it's got all those prefixes. Varicella zoster virus VZV, human herpes virus HHV, and the numbers after it. CMV shares many attributes with other herpes viruses, including the genome, virion structure, and the ability to cause latent and persistent infections. CMV has the largest genome of the herpes viruses. Replication may be categorized into immediately earlier, delayed early, and late gene expression based on time of synthesis after infection. The DNA is replicated by rolling circles. Human CMV grows only in human cells and replicates best in human fibroblasts. So you have the picture up here. All right, this is better. So this is what it looks like. CMV, we've changed, no? Yeah. CMV is a common virus that infects most people at some time during their lives, but rarely causes obvious illness. It is a member of the herpes viridae family. Other members of the herpes virus family causes chickenpox, which I'm sure you've had it one time or the other. In fact, our generation has. Infectious mononucleosis, not likable very at all. Fever blisters, herpes 1, and genital herpes 2, like other herpes viruses, CMV infection. So this is not a very, very nice thing to have. Take a look at the structure. Can you see? All right. It's not as complicated as the other one, no? Right. They may be found throughout the body. HCMV infections are frequently associated with the salivary glands. HCMV infection is typically unnoticed in healthy people, but can be life-threatening for the immunocompromised, such as HIV-infected persons, organ transplant recipients, or newborn babies. After infection, HCMV has the ability to remain latent within the body over long periods of time, and that's what makes it very, very dangerous. Other complications, take a look at this picture, it's sad. CMV mononucleosis, intestinal complications, liver problems, nervous system complications, lung complications. CMV can cause inflammation of your lung tissues, pneumonitis. Complications arise from newborn CMV infections include hearing loss, the child has a problem, eye abnormalities including central vision loss, scarring of the retina, and inflammation of the light-sensing layer of the eye, retinitis, swelling and irritation of the eye, mental disability, attention deficit disorders, hyperactivity, autism, lack of coordination, small head, seizures, and death. Okay, Paro's leaving. All right, her title must be good. Okay. So test for CMV, again, IgG, IgM is asked. Methods are ELISA, ECLIA, using different machines. So we come to the herpes, HSV1 and 2. What is this virus all about? There are 8 human herpes viruses. Also, every animal species that has been investigated also has a herpes virus, my God. What these viruses all have in common is the ability to hide out in the body without causing symptoms and then reappear at a later age. The human herpes virus family includes, and we'll take a look, you can see a little picture on your right hand side. Okay. So what is the virus all about? Herpes simplex virus, cold sores and whitlows on fingers and hands, also half new cases of genital herpes. Herpes simplex virus 2 genital sores, also sometimes cold sores and whitlows. The other viruses cause quite different illnesses that are varicella zoster virus, also called herpes varicella, chickenpox, and herpes zoster shingles, painful. Epstein-Barr virus, EBV, often referred to as EBV, dangerous. Cytomegalovirus, dangerous. Human herpes virus, HHV6 in babies. Human herpes virus 7, HHV7 in children. Human herpes virus 8, HHV, or it can be called KSHV, Kaposi sarcoma, and you know that is bad, skin cancer in AIDS patients. All right. Diseases that can cause herpes simplex is a viral disease caused by both herpes simplex virus type 1 and type 2. Infections with herpes virus is categorized in one of the several distinct disorders based on the site of infection. Oral herpes, the visible symptom of which are colloquially called cold sores or fever blisters, infects the face and mouth. Oral herpes is the most common form of infection. Then you've got genital herpes, known as simply as herpes, is the second most common form of herpes. Other disorders such as herpetic whitlow, herpes gladiatum, ocular herpes, keratitis, and cerebral herpes. Genital herpes is common and highly contagious infections, usually spread through sex. Usually these infection is caused by herpes simplex virus 2, although herpes simplex virus 1, the virus usually responsible for cold sores, may cause genital herpes also. Genital herpes treatment includes medicines to help sores heal faster and prevents outbreaks. So methodologies used in diagnosis, I told you, is ELISA, ECLIA, Roche E411, using ECLIA principle. These are highly specific and sensitive methods, and you have to have the machine for it. And there are other companies also making ECLIA machines. Are these infectious diseases? The answer is yes. Why? They can be spread by skin contact, saliva, sexual contact, and can be fatal in adults and children if untreated. Can these be treated? Yes, with antivirals under doctor's supervision. This can be controlled, and there are various drugs available in the market. The doctor has to recommend it. Okay? We come to more or less the last slide. So here you've got the torch, and you've got this little bug making faces, and that's the last. You see, it says doing now what the patient needs. Next, this is a Roche ending. I've kept it as short and sweet and understandable as possible. I hope you have understood. Any questions, guys? You know of cases? Herpes, toxoplasmosis.
Ma'am, if one has repeated mouth ulcers, should one test for the herpes?
Yes, you can do a buccal smear, and the doctor can ask you. The doctor can ask you for HSV 1 and 2. It's freely available.
The IgG, IgM both are recommended.
Yes, yes, of course. Always.
HSV, only HSV.
Pardon?
Only the HSV test.
Yes, HSV 1 and 2. The herpes test, yeah.
Herpes.
Any other questions, Nazdeen?
So, ma'am, we are using because you say about architect was about ability. Then architect is also heavy, and now civil I'm using that is also heavy.
Can you be a little loud and little slow? A little loud and little slow, Nazdeen? You're walking somewhere. Rohana, anything? Since
I was only testing this when I was working in the virology department. We
tell
had
us.
samples coming to us,
Mm.
but I've seen people having this on their lips. Many of them were always
Yeah,
suffering.
yeah,
They have it on their
corners,
lips.
yeah.
Yeah, always on the corner. I also had it once.
Mm. And herpes is a little
Ma'am,
difficult to
I
cure. Bolo. Bolo?
sorry, Rohan, you please carry on.
No, I was just saying that I also
No,
had
because
it when I was
I know
testing.
mom, my mom developed it, and it only comes halfway to trunk. Only halfway. And she had a big band, nearly 4-inch band, red band with these typical little beads. And she said she didn't tell me, she tried to avoid it, and then she was in a lot of pain, I could see. I sent her immediately to my uncle who's a doctor, and he put her on 2-3 injections and some creams, and she recovered very fast, you know. And she was told to keep changing the sadras because we Parsis wear that malmal ka sadras, and keep it dry as far as possible. And she recovered very quickly. I don't know what shots Dr. Ellis gave her, and the cream and some tablets, and came out of it very fast, very fast. And herpes very it just takes half. Trunk ke pass was itna here. It doesn't go to the other side. She had a 4-inch band, and in the beginning she was very painful because it affects the nerves. A lot of pain. Then eventually I pushed her. I said, "Go see Dr. I said, "That mom, this looks like herpes," and right enough.
I had one case where it was the herpes was spreading on the face,
Mm.
and it can spread when you are eating food with somebody also. When you are sharing some of the food or some,
Possible, yes.
it can spread
Saliva mein hai na?
also.
Yes.
Yes. So suddenly it developed on my face, and he had got it from somewhere in the other person.
Mm.
So we couldn't understand, and then when we traced it back, it was to the food, found it to the food, how it could be contagious so much, so fast. Also, the manifestation is very fast. Within 6 hours, it will develop. It's very fast. So and here we are using about architect, LMC, about LMC architect, and this now I'm using humans. Humans also helpful profile. So
Mm.
within 20 minutes it will be IgG, IgM, it will be ready. But the only problem is pre-analysis when the doctors want to order either it is one IgG, it is two IgG, IgM combination. There are totally six. Out of the six, which one the doctor wants? So mostly time goes over there collecting history and everything. If it is positive,
Yeah, but the
and then
crux
again
is, I've always said that taking history on the patient is very important. And I'll tell you, like I think you were my student, I said that 90% of patients will tell you what's wrong with them. 10% will tell you, mind your own business.
no, this country they will deport now, so they don't tell. In this country they will deport, so they will not tell. We have to get it out of their mouth.
Mm, 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 mm.
Some things are privacy, and a lot of privacy in Kuwait.
No,
Yeah, privacy.
but see, we as techs have to maintain that integrity, na? When a patient tells you something, कि भाई ये हुआ, they've been around. You can't go around broadcasting. Oh, his VDR was reactive, or his herpes was reactive. You can't do that. You're sworn. You're
What
sworn.
happens is, ma'am, we have to report these inconsistencies to the system,
Mm,
case, per person,
mm,
per case. Now what happens is previously people were defaulting, medical centers were defaulting. So DSA made a rule that all the reports have to pass through a system where they will be able to see our report. And
mm,
today morning only we got one notification that three evidence ID card numbers were wrong. So they are actually monitoring everything which goes through their system. So nobody can escape from that. Nobody can default also. Now previously it was manual. We had to write all the manual count. Daily 1, 2, 3, 4 like that, 1, 1, 2. Now no need for us to write. Everything goes to their system and AI is collecting in their system all the counts. So here the government is very strict on that. We cannot escape that. And there are 14 divisions we have to inform the government. So they will take care of that patient, they will take them into custody, quarantine, treatment, and then if it is very much infectious like HIV and all, then they will be reported after confirmatory tests and all. So that is the complication over here. So that's why each sample is taken as infectious, already known infectious. You have to, like level 2, biosafety, PPE, everything has to be done like that. So by default only we are saying like that.
Okay.
Yeah, but when I was also working in virology, except for HIV and all the others, we knew, who the patient is. We could give them whatever is required, and the results could be said. But HIV and all, we didn't even have the names. The names also were not revealed to us. Always initials. And even when I was doing HIV sequencing, we were not allowed to, know who the patient is, what medication he is on, that way. It was too private. Of course, anybody else besides a QAP, they are. Now it is very less. HIV is very less.
Yes. Inside the lab, the worker could come to know the patient identity. The laboratory, as a referral laboratory, the patient identity we don't come to know. But only thing we come to know the result which we have to inform our client. And then they will inform the DHA. But now, overall also, all hospitals, medical centers, laboratories, no need to offer all this manual work. It is getting screened from the government side only.
It is centralized, yeah.
Yeah, from the news connected to our news, and government is only tracking it like that.
Okay.
Same like
Interesting.
radiology also for the infectious